this kind of. In this case, only mannitol can be used. Okay, this is just an example of two-stage fermentation. Firstly, mannitol is here, but here we inoculated the gluconolactic oxidants, then increase the mannitol, fructose. Then here we inoculated Aurantiochytrium, then they grows Aurantiochytrium and then produce DHA or astaxanthin and carotenoids. So this is very good for this solution. Okay, mannitol is good, but the other main component is arginate. Arginate is maybe you know that uronate and gluconate containing sticky polysaccharides. So this is also very difficult to use. So we don't have any good enzyme to hydrolyze or conversion to other materials. But we also the same notion that two microorganism system. So our team searched very good and found that this monotonous bacteria really very good. If you treat the arginate medium by this microorganism, then Aurantiochytrium strains can be very good growth. But if not, they don't do. So this monotonous microorganism may produce something from arginate. But firstly, we didn't know when we checked what it is produced. The answer was the organic acids, especially acetic acid. Acetic may be a very good substrate for this microorganism. In the case of glucose, very rapid assimilation and a very high amount of lipids compared to that acetic acid, it's not very good, but still even practical to use. Slow assimilation, slow consumption, and the slow production of lipids, but still we thought that this is good. So also acetic acid. Okay, so I mentioned that the meta-fermentation. This is meta-fermentation system, and Nakashima Sensei every day taught. Then he mentioned acetic acid, intermediates meta-fermentation system. So we found alpha. I noticed that acetic acid can be supplied from this system. Okay? In the meta-fermentation, usually complex microorganisms hydrolyzed or acidogenesis by this kind of microorganisms at the first step. And the second step, higher organic acid, except the acetic acid, converted into acetate or hydrogen carbon dioxide by this kind of. So in this case, microorganism called the Clostridium or Acetobacterium and the Morella called acetogen. So because they produce acetic acid, so and after that, methane was formed by this kind of methanogenesis. Something. Okay, so I found that if acetic acid can be supplied by this system, then Aurantiochytrium can utilize acetic acid to produce lipids. Okay, so then I found that the kombu or macroalgae is a very good substrate, but still very limited usage. So I thought that CO2 can be a new material, raw material for production of lipids by using this kind of system. So we selected acetogen as a first microorganism that converts CO2 into acetic acid. And after that, acetic acid converted by Aurantiochytrium to produce lipids. Okay. So acetogen has this kind of ungual pathway or acetyl-CoA pathway. So they utilize 2 moles of CO2, and one of them are converted in the nature end, and the other is packaged. Okay, so they produce acetyl-CoA. Then after that, they are degraded into acetate, ATP. They obtain ATP, but that is consumed by this system. Okay. So in terms of energy production, there's maybe no net zero, but acetate is excreted. Then some of those, maybe 10% of acetyl-CoA converted to any components constructive violence, violence in the cell column. And the other 90% are excreted as an acetate. So they can utilize by Aurantiochytrium. Okay. So we check if that is really true or not. So we selected acetobacterium as an acetogen to convert CO2 into acid. I didn't mention that the reduction of CO2, we need maybe here you see 4 moles of hydrogen. So that's also a problem. But if you can supply hydrogen as a too, then that's good. Okay. Here, acetobacterium would be the first microorganism to convert CO2 to acetate. It grows here. This microorganism is anaerobic bacteria. So this is different from the gluconobacter stem. That is aerobic. Here, anaerobic. So the condition is different, but they produce also acetate. So in this case, almost 2-3% of acetate is produced here, and then directly inoculate Aurantiochytrium into that medium. Then we found that the growth very good, and the 60% of cell can be lipids. So this combination is very good. Then we believe that this can be the solution to reduce CO2 from societies. Okay, we also have a pattern here. Then we call this system as a gas-to-lipids by process. Maybe you know that gas-to-lipids by process, but this case, lipids, because acetate can be also lipids, liquids. But I mean that very focus in the lipids. So gas-to-lipids by process may be a good name. Okay. So we propose that this system is a very good solution for reducing the carbon dioxide from the industrial waste or any activity, human activities. Also, most of the biomass can be a substrate of the system because acetogen can utilize this kind of many kinds of biomass as a material, or they can be classified into the CO2, and they can be a substrate, or even this kind of power generation, hydrogen generation, they produce a much higher amount of CO2. So most of the this kind of biomass or any underutilized materials can be substrate, and they can convert into acetate as an efficiency of up to 90%. So very good. Then Aurantiochytrium utilizes that acetate into this. So this system can be good. Okay, then I added this CO2 as a new substrate for this conversion system. Then conversion of various resources by other organisms. But one of the this successful system applied to any other biomass. Okay? And also I talk later about the genomic editing to breed this microorganism to produce much higher efficiency. Okay? But anyway, so I talked about the CO2 utilization. So maybe you know that the CCU or CCS So carbon dioxide capture and utilization or storage. Nowadays, a solution to that reduce the CO2 or from the industrial factory or power generation system. So nowadays, people say that the carbon dioxide is a resource. This is not a waste, not a resource. So in my the knowledge, this plant artificial photosynthesis can be a very good solution to reduce or consume CO2 to make something very good. In that case, similar to the plant photosystem, catalysts are used, maybe chemical or metal catalysts are used to hydrolysis to produce hydrogen. Then carbon dioxide are reduced by this hydrogen to make olefin or something. And that is very similar to the plant photosynthesis. So artificial photosynthesis is very good. But this is, very similar to my case. So because catalysts are metal or chemical catalysts, but in my case, the biocatalyst. So this is very similar. And in my case, hydrogen is used to reduce the CO2, but also oxygen can be utilized by Aurantiochytrium. So there's no issue. Okay, so I talk about the problem. So this picture shows the primary energy composition in different countries, and then you see here the Japanese got the English, petroleum, natural gas, and coal. So these are the, I don't remember, but fossil energy. So we can here the line. So these are still 80% or so. This is 2022. So this is not very old. And nuclear power or hydroelectric or even renewable energy, it is still increasing, but still 20% or so. So still we rely on this kind of fossil fuels. Summer power generation. Also, especially this kind of, a very big producer, China, USA, India, Russia, very negative to carbonization policies. So that could be a problem. So in Japan, very small amount, but a very, we are trying to reduce this kind of things, but we need to ask them to do something like this. Okay, anyway, so if we can introduce very good way to reduce CO2 or consume CO2, then they may use that system. In Japan, so there are many trials to reduce, to utilize fossil fuels, like LNG, natural gas, and then coal-fired power. So in this case, there are, sorry here the Japanese, but 2020 and 2030. So nowadays here, and we are seeing here that the IGCC and the IGFC, which are trying by the power generation company that reduce the utilize of the coal to or natural gas to produce higher amount of electricity. That is done. That is. Yes. Then that is done. Actually, Hiroshima University is here in Japan, and then about 1 hour by using car and ferry ship to reach this Osaki Kanji Island. So here, you see here, Osaki Fuji. The Fujian is the project to efficiently produce the next generation power generation. And also they are trying to carbon recycle because using this system. So they are primarily positive oxygen from integrated coal gasification combined cycle to produce electric power. So they utilize the steam and also gases by gasified by doing the oxygen draw. The coal is stored energy source. Then they produce a high amount of energy by high efficiency. But also they recover the CO on H2. This is synthetic gas. So this converted by the ship reactor by adding the H2O to make a CO2 and hydrogen. This is very stable compounds. But the CO is very toxic and unstable compound. So they convert CO2 and then hydrogen. And then recover only CO2 and hydrogen. That is the CO2 capture system. The hydrogen returned to this system and then utilized to make electricity. But we see here the CO2 recover at a very high concentration, more than 99 something percent. Then they can utilize for something, utilize CCU. Then we are okay. They say that okay, people can try to utilize this CO2 to make something good. So we propose a system. Then finally adaptive to make a very good money. Then this is R&D and demonstration base for carbon recycling system with also production station. The Osaki Kugen is the next here. Then our system built here. And many projects are adapted, and then our case is here. So I don't talk much about this, but in my case here, but the other is anyway, the RJ, green RJ is here, and so the concrete system is here. Okay, please see this website if you want to see. Okay, my project is the development of a gastrolipid management system. The picture is very similar to acetogen and this kind of Aurantiochytrium. Acetic acid can be an intermediate. Then we again need the sustainable H2 because the cost is very big here. So we need a cheap hydrogen. And also we also need the CO2 at a high concentration. Okay, and this system, this is a very proud system to do this research. Okay, and yeah, I will talk about the merit of this. Now, I put here. Because these are not doing not carry out photosynthesis, so they don't need essentially the light irradiation. Okay, so that is very good because we can use the tank, which does not have any like irradiation or something. So that is very good. And that is also the merit to make a high concentration, high cell concentration, because they don't need to capture the light. So yeah, but besides this one, the green algae need to capture the light, so they can be low concentration. They should be a low concentration. So they can be a very big culture system to make high amount of algae. Okay, so that is very merit for this system. And so, yeah, this is our facilities for tent. Then inside this, we have an in-house laboratory and the plant system to do the two-stage fermentation system. Okay? The first one is this is acetic acid fermentation technology system, using CO2. But in this case, there is no propeller or something because we don't create the spark. Spark maybe a burst by because they're using hydrogen, so high amount of hydrogen here. So the bubble is incorporated at the bottom of this tower system. So here 40 liter or something, then I 3 or 4 liters. Then the CO2 and the H2 are incorporated in the system, and the bubble is going up. And during that, CO2 and H2 are solved in the water. Then they are consumed by acetogens. Okay? Then they make acetic acid, and then we recover that by filtering this. Then acetic acid solutions here. And the cells are returned to the system, and then utilize this. But some of the CO2 and H2 are escaped from here, then we recover that, and then measure the concentration of the acids, and then use again. Okay? Then these are one is huge acids here, and then 100% gases here. So gas is still, there is no way to escape. So we can utilize carbon dioxide and hydrogen at very high efficiency. As I mentioned, 90% of carbon is converted in acetate, so conversion ratio is very high. Okay? And here is an example of the gas circulation rate and the fermentation rate. So I don't talk much about this, but very stable fermentation here. And we see here the acetic acid concentration. So for example, 80 gram per L, so around 2% or a little higher than 2%. So I will mention about this later. Remember this number. Okay, anyway, so in this way we can do the acetic acid fermentation at high efficiency. Then once we obtain the acetic acid containing products here, then we utilize that for the lipid fermentation. We use 2 tanks, but not necessarily. One tank is okay, but in this case, the cell proliferation tank is here. Here is the nitrogen concentration is higher because the cell grows rapidly. Then the second fermentation is for the lipid accumulation, because in this case, the carbon concentration will come because they need to make lipids. Okay? So by this way, also efficient condition, higher production condition is done, and then we recover the cell to extract the lipids. Okay, so this is just not very good data, but here very stable continuous culture conducted by this system. But here we have a problem here that dissolved oxygen air, because the Aurantiochytrium should have oxygen to utilize metabolism. So we include some aeration system, this is kind of very nanoparticles to increase the dissolved oxygen. Okay? So then we increased some lipid yield versus acetic acid, but 24%. 24% of acetic acid is converted into the lipids. And maybe 30% of them be protein or something in the cell, and the other is exhausted. So we also thinking that exhausted CO2 can be recovered and then be utilized for the acetic acid fermentation. So that system is okay. But still, we calculate that to maintain these numbers, so we need the acetic acid concentration for the medium as a maybe 5%, 50%. So, 5%. So I mentioned that acetic acid fermentation need around 2%. So we need to concentrate acetic acid to maintain this kind of system. So Nakashima sensei discovered that this kind of the polymer can be very good to concentrate acetic acid. He will mention it. Okay, this way, so we have done in 4 or 5 years using this process to obtain this kind of numbers. The numbers are still lower than a year ago, but we are still doing this for improving the system. Okay, so this system, as I mentioned, very high efficiency, and also dark place is okay, so we don't need the light. In Japan, here maybe sunlight is very strong, that's good, but in Japan there's no such a strong sunlight, so we need to use electric power to irradiate, right? But in this case, there's no need. Okay? That's very good. And then this, because of that, microorganisms, maybe our system, maybe some good point is there. But the how we obtain cheap hydrogen. Okay? That's also the problem here, that they don't need microorganisms. Okay, so but the problem is that acetic acid is not still very good substrate compared to the glucose or any the kind of saccharide, because the saccharide is very good. But in the major pathway, the glycolysis make some ATP and the reducing power. So that's very good. But acetic acid will not have such a energy production process. So we need to see what is necessary to improve to utilize lipids by using acetic acid. So we conducted a metabol analysis using this kind of spectrometry or something. Okay? Here we see heat map there. The 2-3 clusters, I don't mention much about this, but compared to the glucose acetate has a very low level of this cluster 1, cluster 2, cluster 3. Many compounds is very low. Okay? So mapping this to the pathway, okay? As I mentioned, glycolysis make this kind of pentose phosphate pathway is this kind of reducing power, or glycolysis make ATP. In this graph, blue one is glucose and red one is acetate. So if you use the acetate, there is no this kind of things here, or because pathway is very low activity, so they don't make efficiently the reducing power. Okay? So the one problem is the deficiency when you reach the point that fatty acid synthesis. So using the acetic acid, so there's no not very bad, but efficiency is still lower than the glucose. The second problem is here malic enzyme that produce also the reducing power, but here is not maybe more data, but gene expression is very low in the case of acetate. That is the second problem. The third problem is here in the TCA cycle, and the bypass using the glycolysis cycle. So this make this kind of a halfway to around the TCA cycle in case of acetate. So in this case, there is no chance to make this kind of reducing power. So there's many problems when we use the acetate. So we need to solve that problem by breeding the microorganisms. So we tried something. I don't know what's happening. Okay. Anyway, so if you use acetic acid, so ATP is consumed by making the acetyl-CoA. Compared to the glucose case, there's many pathways, and then eventually come to the acetyl-CoA that produce many ATP or reducing power. So that's a totally different. And then acetic acid is not very good in that motion. Then this consumption of ATP makes ATP production, and then this ratio is increased because ATP is consumed. Okay? So in this case, the SNP1 genes, this is AMP-activated protein kinase. That is, the master regulator of the system. Then they activate the beta oxidation in peroxisome and mitochondria. Because the fatty acid is degraded by beta oxidation system, they make ATP and release power. So they compensate by activating this beta oxidation system when this ratio is increased. Okay? So we see this kind of APOX and the R1 genes, or even this kind of system, and also SNP1 gene expression or any metabolites production, research by metabolites. Then we found that destruction of these genes, ACOX1, ACOX2 genes destruction makes increase of fatty acid compared to the wild type strain. So this means that these enzymes are very important to reduce the beta oxidation activity. On the other hand, SNP1 is a very drastic change. So, and in the glucose system, the low concentration of glucose, batch culture, they make some good growth, but maybe similar to wild type system, but flow culture, which is a high concentration of glucose, they suppress the growth because SNP1 is activated by high osmotic pressure, so they suppress the growth of those. And but this is glucose case. This is acetate case. They also suppressed here and there the high or low concentration of acetate. There's no different. They totally suppress their growth and then very low level of lipid production. This means that the SNP1 to 1 is very important to utilize acetic acid. Maybe they are essential for the utilization of non-glucose carbon stores. That is found in the yeast. So we see the yeast characteristic to solve this problem. Okay, that is one topic, and the other final topic is the carotenoids. As I mentioned, Aurantiochytrium make a very high producer of carotenoids, especially xanthophyls containing such as canthaxanthin and astaxanthin. Astaxanthin is very famous, and this kind of red compounds, then very strong antioxidant activity. Okay? At this moment, the chemical synthesis or Haematococcus algae is a producer of this astaxanthin. But there are some problems. So to solve this problem, Aurantiochytrium can be a good alternative producer. We breed by using the chemical mutation system to increase the carotenoids and astaxanthin. The very reddish color is formed. Strength. And also, we also found that the enzyme called CRT-IDY is a very key enzyme to produce this kind of carotenoids because they convert GTPP, which is no color compound. Then they produce this kind of lycopene. Lycopene is carotenoids in tomato, so this is red. And this is yellow. Carotene is a carrot. Okay. So this kind of carotenoids are produced by this CRT-IDY in this microorganism. So these three enzymes are combined. So if you disrupt this enzyme, then GDPP cannot be converted here. So the red colonies becomes white colonies. So this conversion is very good to see disruption of this gene. So to do to take this advantage, we applied genome editing system to Aurantiochytrium for the first time. The CRISPR-Cas9 system is very famous, and you know that this enzyme Cas9 is guided by the guide RNA, then cut without strand of DNA. Then the recovery system, and then cut and recover. Then eventually they have a mutation here, and then after that the mutation is fixed. Then phenotype is changed. So by using this system, we try to disrupt CRT-IBY to convert this red colony into white colonies. Okay? We found that some white colonies are there, and then we found this colonies disrupted the CRT-IBY system. Then this is first time to see the Aurantiochytrium to apply the genome editing system. We also tried the plasmid incorporation system, and also the Cas9 enzyme and the guide RNAs are combined in vitro, and then they are incorporated in cell, and then the red colony becomes white. So in this case, we don't incorporate any genes in the genome of the Aurantiochytrium, so it's very good and a very efficient system. So we now utilize this system to disrupt any genes, genome-wide system, to check if which genes are very effective to increase the lipid production. Okay, in that way, so conversion of various resources like other organisms, the CO2 can be very good substrate, and the breeding of the genome edit becomes very good, the result. Okay, so by doing the collaboration with Shannon or in this university, I myself need a very good, very cheap acetic acid for lipid fermentation. Also, of course, the acetic acid fermentation will need the other resource to the other way to produce this kind of system. Okay, thank you. And then, yeah, this is announcement for our collaborators, and then we use the okay, thank you very much for your attention.
Thank you very much, Mr. Aki. We have some time for question and answer. So I think it's a good chance if you have any questions or you would like to know more about research. Anyone? Yeah. For me, I would like to add. Oh, okay.
Thank you very much. I'm Salvarat from Faculty of Science. We in the Biochemistry department. So in my lab, we also work with Aurantiochytrium, but we don't get the red color yet. So yeah, but we work a lot on carotenoids from Aurantiochytrium. So today your presentations make like inspiring for us. So I have questions about the because we work about DHA, right? Lipids and DHAs, and we found high contents of those molecules or things and carotenoids also, but there are like different conditions to induce the production. Like if we would like to produce lipid, we're gonna do these conditions. But if we need like more carotenoids, so we just switch to another condition. So I have question that when you make like upscaling productions like fermentations, I'm not sure is that secret or not. You have like a specific condition to induce the productions of those like also DHA, lipids, or carotenoids. I mean, I think it's something maybe really important for us.
Okay, thank you for your questions. And then, actually, that's good question, and that's what I want to know. So I cannot answer essentially, but we see that lipid production, I mean DHA or something, and this is the very first half of the calculation. Then carotenoids are the second half of the calculation. So we can, I think there may be the switching between the first half and the second half of the calculation. So maybe I think the stress, culturing stress may induce the production of carotenoids. So I'm trying to find what is the stress. Maybe the concentration of the cell or reduce something, oxygen or demand or something. And stress may induce the carotenoid production. So if I can find, or you can find that, then you switch on the stress at the first, then maybe increase. Or you want to switch, right? So yeah, maybe yeah. Oh yeah, yes. Yes, but in this case, lipid production system is a very fatal, and it cannot be reduced. So I think you should think about I also think about the stress inducing or any signaling system. Yeah, that's my answer. Possible answer. There's no secret.
Is there any more questions?
Hello, Professor Aki. I'm Sorada from here. If you make hydrocarbon or fatty acids, how homogeneous is your product? I mean, do you get one fatty acid 70%, or you get like half of something and another half something?
You mean the fatty acid composition?
Yes.
So typically, yes, the permitted is 50%. DHA is 30 or 40%. The other is other 22 carbon polyunsaturated fatty acid, like docosahexaenoic acid or something. So totally 30 or 40% polyunsaturated fatty acid. It's maybe two kinds, only two kinds. The permitted is high. So very simple. Yeah.
And normally the downstream process of these two are still using the extraction, or you have the other ways to get it out?
Get it out what?
From the cells. I mean, from
yes,
the broth.
There are many ways and some secrets in the company or something.
Okay.
We actually don't know the current situation, but we utilize usually the organic solvents.
So, okay. Thank you. Yes.
Any more questions? Maybe one more? Two from like a bump.
Thank you for sharing with us. It's just an interesting study. I think you laid a good foundation to capture the CO2 by your Acetobacterium species. I'm curious, is it possible to add like some biomimics for samples like a soybean pulp to add for the substrate, and then with the produced acetic acid, and then because I think it's quite challenging to find like organism to utilize the acetic acid alone, because not a lot of organisms, because actually I'm working with fungi like aspergillus and something like that, and we still need like a glucose or something like that. So do you think like is it a good idea to mix the produced acetic acid with some biomass and produce filter fermentation with other microorganisms?
Yeah, of course. I mentioned here specifically the acetic acid bacteria, the Aurantiochytrium, but my patent shows that many other suppliers of acetic acid, and I mentioned the acetic acid is one example, the substrate for this lipid fermentation. So there's many the first organisms. But in this case, CO2 utilization is a very big issues. Then I think this system can be the biggest one for my system, but other microorganisms can be also utilized. Is that your question or?
I think he meant that for second fermentation of
Yes.
acetic acid, if the bacteria, if the organism doesn't like to eat acetic acid, then we add biomass into the fermentation to help towards the final process instead of that acetic acid.
Other biomass, what kind of biomass? Sugar?
For example, like a soybean pulp or cassava waste.
Oh yes, we use something like that, the protein, because they need the protein. Yeah, I didn't mention about the composition of medium, but we utilize such a process.
Yes,
They need actually.
Thank you very much.
All right. I guess okay. One last question.
Good morning. I'm Kasidik from Chemical Engineering Department. I'm just wondering whether, from what I know, carbon dioxide and hydrogen gas is are not good water suitable compounds. So I'm just wondering how much, or have you ever assessed how much of those compounds remain unused or released into the environment? So because I believe that if you can increase the amount of the hydrogen and carbon dioxide solubility, and you can get more acetate, so you can get more of the subsequent product. So please enlighten me how did you deal with the water solubility issue of hydrogen and carbon dioxide?
Very difficult question. Maybe I cannot get your point, and then that is really Nakashimata's process. We answer.
I think you
No.
have the reactor.
Oh yes.
That bubble the hydrogen from.
Oh yes.
So it has a lot of chance to be converted. Is it? Okay.
Chance to be converted to acetate.
Acetic acid.
Oh yes, of course yes. And what?
And you said the US 90%.
Oh, that depends on the speed, the flow rate or concentration stuff. So the optimization in that condition will be done in this the project. He will mention that.
Okay, let's join me to thank Professor Aki once again. Yeah. Next we have Professor Nagashimada from Hiroshima